very warm hello and welcome to this latest Science Custom podcast, created in partnership with Bold, the blog on learning and development. I'm Sean Sanders, Director and Senior Editor for Custom Publishing at Science, and I'm very pleased to invite you to join me for this series of podcast interviews with outstanding researchers who are attempting to make positive changes in the lives of children and adolescents by seeking practical solutions for a complex world. Apart from this common goal, they are also recipients of the prestigious Klaus J. Jacobs Research Prize, a 1 million Swiss franc grant awarded by the Jacobs Foundation that recognizes exceptional achievements in the field of child and youth development. My guest today is Dr. Michael Meaney. Michael is the James McGill Professor of Medicine at Douglas Mental Health University Institute of McGill University and Director of the Translational Neuroscience Program at the Singapore Institute for Clinical Sciences. His primary interest is in studying how experiences in childhood, particularly the type and amount of caring provided by mothers, can be carried in our DNA into adulthood to shape brain development and later health outcomes. Michael, thank you so much for joining me today.
Oh, it's a pleasure, Sean. Glad to be here.
So, Michael, for many years, you've been studying the effect of caregiving in childhood. What I find most fascinating is how the impact of negative events in childhood can be recorded by our bodies through modifications to our DNA. Can you describe to our audience what your research has taught us?
Yeah, I think your audience is probably now very familiar with the idea that early life events, the experiences of early life can shape the connections in our brain. I think our contribution to this was to actually show that these same early experiences can actually modify our genome. I want to be very clear here. I am not talking about a change in the letters that comprise the sequence of our DNA. But what we're talking about instead are actually chemical modifications that are imposed on the DNA, the software, if you will, that determines how the DNA operates within any particular type of cell. And what we've shown is that this software, these chemical modifications, can be modified by early environmental events, including early life stress. And in doing so, they can actually modify the operation of the genomic hardware. Now, this, I think, probably sounds a little outlandish, but it's actually part of our normal development. I mean, if you think about it, one of the real mysteries of life is that our bodies, over the course of development, produce over 200 different types of cells. And all of those cells have exactly the same DNA. So somehow that DNA has to operate very differently in these cells. And these chemical or epigenetic modifications are really the answer to this problem. And so if you look in cells in your liver, your muscle, your brain, these epigenetic modifications comprise the software that determines whether or not that cell is operating as a liver, muscle, or brain cell. So that even though each cell has the same DNA, it's read very differently to produce different types of cells. Now, our contribution was to sort of take it to another level. So we weren't interested so much in why a liver cell was different from a brain cell. We wanted to know why the liver cells or brain cells of one person might differ from another. And in the course of doing this, what we found is that early life experience can actually produce similar types of epigenetic modifications that can then actually determine why your liver or brain cells operate differently over the course of the lifespan.
I've always thought that people respond to stress in a mostly similar way. Is this not true?
Actually, no. There's remarkable individual differences in individuals in terms of how they respond to stress. And in part, these individual differences are determined very early in life, and they can be highly adaptive. I mean, we need to think that our stress response is there to serve our needs under conditions of duress. Children living under conditions of violence, for example, are very well served by being highly sensitive to any threat and being able to mount a vigorous stress response accordingly, including the release of stress hormones that will help them form memories of these threatening situations to better avoid them in the future. Simply put, these children need a more vigorous stress response. And so when we look at early life, the types of adversity that children can experience in early life can actually enhance the sensitivity of their stress responses. But again, we need to keep in mind that this can actually be highly adaptive. It can help the individuals survive and deal with the threats of trauma in their lives. The problem, of course, is that the chronic activation of these stress responses can also take a toll. So it can lead to a greater risk for health outcomes in later life. And this is one of the reasons why, for example, we think that early life stressors promote the risk for so many different forms of illness over the course of the lifespan.
In your research, do you see any difference in boys or girls in their response to maternal caregiving and also their responses to environmental stresses later in life?
Yeah, this is puzzling because it took us quite a while to land on this. And I mean, the global but it's a really rapidly emerging story, is that, yes, there are substantial differences in the way boys and girls respond to both early life stress as well as to the caregiving of parents. There's an incredible need to be very precise in the way we phrase this. In general, what you see when looking at kids that are experiencing early life adversity is that both boys and girls are affected. It is not the case that one is more responsive or sensitive than the other, but they're affected in different ways. Girls tend to be more affected in areas that are social and emotional. Boys tend to be more effective in areas that we would think of as cognitive, learning and memory, for example. But again, we really need to respect the nuances. So, for example, yes, there are gender differences when we look at the response of boys and girls to family function and parental care. But if you look at other factors, for example, like poverty, you can see a very different pattern. And so our colleagues, of course, would also point out that there are also sex differences in the impact of paternal care, the care of the father for boys and girls. I think we get to the point where when we want to take this story to completion, we're really going to have to map it out very differently for boys and girls. There are different ways in which they're affected, and those influences of early life stress lead them on different pathways, in many cases, to different forms of health problems.
Michael, in your work, you focus primarily on maternal caregivers. Have you also looked beyond mothers to fathers and extended families as caregivers and what impact that might have?
Yes, I wish we'd done this more, to be honest with you. We've been a bit overly focused, but I can certainly summarize what is well known in the field. It's a critically important factor. There's research going back to the 1950s, wonderful studies showing, for example, how members of an extended family can turn out to be so highly protective, even in the midst of families that are experiencing severe dysfunction. There is a remarkable capacity for what people refer to as buffering effects that indeed occupy the role of both parents, but as well as extended members of the family, even beyond members of the family. So, for example, in cross-cultural studies, we're used to the idea that extended families have many caretakers, grandparents, for example, as would be the case here and often in Singapore and other parts of Asia. Where we're also learning in Western countries is that highly functional daycares can also serve to buffer children against the effects of family dysfunction and other forms of early life adversity. So children are remarkably good at being able to take advantage of the forms of social support that surround them. And these can be very effective in buffering the children against the effects of early life adversity.
So a lot of the work in this field seems to have focused on negative drivers like stress and depression. Are there any studies following the positive aspects of caregiving?
Oh my goodness. So you've landed on what was really one of the major irritants of a wonderful research fellow, Desiree Fouad, here in Singapore, who was not impressed by our relentless emphasis on parental depression and negative forms of caregiving and wanted to look at positive maternal mental health. And so she developed a measure by which she could look, for example, at how mothers do very well and are very calm in dealing with the stressors of their life, very self-confident. And what Desiree able to show, I think, was that, first of all, these forms of positive maternal mental health lead to very positive forms of caregiving. And to some extent, they have very distinct. And I think we need to keep in mind, as she would remind us, that positive maternal mental health isn't just the inverse of negative mental health. It's actually quite separate. And the two are modestly related. It's a very distinct influence. And positive maternal mental health leads to very specific forms of maternal care, those that we would think of as authoritative, where parents are providing children with sensitive instruction and structure and are very sensitive to the needs of the child. And these forms of positive caregiving that are promoted by positive maternal mental health also have very specific outcomes. They tend to be highly influential with respect to social development and language development in children. So I think she's given us good reason to start thinking more in terms of the positive aspects. And in a sense, if your listeners wanted to think about this, when you look at parents who are high in positive maternal mental health, what they can best be characterized as doing mindful parenting.
Interesting. Now, I wanted to come back to what you were talking about earlier about the epigenetics that are involved in this software that you mentioned. Now that we have a better handle on how the body stores the information about stress and abuse and also the positive side, is there any way that we can modify these records or erase negative effects?
Yeah, and I want to be really clear because this is a critical question, and I'm sure very much so for many of your listeners. Erase, I would say no. Modify, absolutely. I think when we look, for example, at the term childhood trauma or childhood adversity, this is a bit misleading. I'm sure individuals who are listening to this podcast who have experienced childhood trauma know that its influence is far from limited to the childhood period. These scars are often an everyday influence across the lifespan for individuals. And so people are reliving the moments, the trauma, the fears, and the effects. So they may have their origins in childhood, but their influences operate across the lifespan. But then we need to think about the fact that there are ways to modify these particular effects. There are interventions. For example, when we look at what are the most common forms of psychotherapy, cognitive behavioral therapy, interpersonal therapy, these therapies are very often designed to modify the way people are responding to the threats and challenges in their everyday life. And so in this case, the effect is not to erase the impact, but it's to modify its impact in the way we're continuing to deal with the challenges of our life. Mindfulness, for example, is another way, which is in many ways synonymous with these various forms of what we call talking psychotherapies. So in essence, what we're doing is we're trying not to erase. This is not going to happen. But what we can do is to modify the way in which the experience of trauma is influencing people's interaction with their everyday world in an ongoing way. So I think, yes, there's every reason to assume that we can modify these effects. The scars may be there, but they need not necessarily be deterministic with respect to the long-term health outcomes.
I'm interested to know, Michael, does your work help us understand why some children seem to be more resilient to adversity than others?
Yes, it does. And in fact, actually, Sean, this has become kind of our primary focus at this particular point in time, because you cannot study the effect of adversity on children without becoming amazed at the capacity for resilience. There are individuals who have experienced remarkable levels of adversity over the course of their childhood into adolescence, and yet continue to function well, seemingly without huge effects. So there's two major influences that we know that could explain resilience under such conditions. The first refers to what we spoke about before. These are the social buffers, the extended family, people, grandparents, aunts, uncles, neighbors, individuals who are always there for us, who served to help us deal with and to buffer us against the effects of these traumas in early life. And so social buffers have long been known as a source of resilience for individuals. And of course, we hear in biographies, telling of these sorts of influences time and again. Perhaps what's emerging is a difference in the way we think of genetics, particularly when we think of genetics that may predispose us to certain forms of mental disorders. So like everybody else in the business, we set out to try to find heritable genetic variants that could predict a risk for later mental disorders, depressions, addictions, schizophrenia, anxiety disorders, etc. And in fact, what we've come to understand is that rather than genetics actually determining the risk for a specific mental illness, it seems to be operating very generally. It seems to be just making us in general more vulnerable for certain forms of mental illness. And it seems that in many cases that the way in which these genetic influences are operating is to unnecessarily exaggerate our responses to stress by increasing our sensitivity to environmental influences in general, and particularly to those that are stressful. This is actually, I think, an emerging idea. It's one that helps us to understand why, for example, individuals who are born into families rife with a history of mental health problems can be at risk for so many different forms of problems, even different from ones suffered by their parents. And again, so the notion here is that these individuals have inherited not necessarily a predisposition to a specific form of mental disorder, but rather to an exaggerated response to stress that then increases their risk for many different other forms of disorders. Now, the interesting component to this is that if indeed this idea emerges as correct, then these same genetic influences that render individuals more sensitive to their environment may also render them more sensitive to the positive influences in their lives. And so again, I think the point here is to really start to understand the nuances here. Rather than thinking of such individuals as being simply at greater risk, we may think of these individuals as also being more vulnerable and sensitive to interventions in early life that could have positive outcomes.
I have just one final question for you, Michael. How will the work that you're doing have a measurable impact in the lives of children, and how can this be assessed?
Yeah, well, this, Sean, is exactly what took us to the topic of resilience. I suppose our strongest motivation here is that when we look at mental disorders, all of these illnesses show a very early age of onset. Every common mental disorder has a peak age of onset at about the time of puberty. So unlike cardiovascular and metabolic disease, we do not have a very big window for intervention. We need to be targeting interventions to childhood, preferably early childhood. And the huge challenge at this point is that our ability to identify which children in early life are at risk for later mental disorders is extremely poor. For the most part, the only way in which we do that these days is by simply looking at children who have been exposed to any form of environmental stress in early life. And as we noted in the previous question, there's enormous variation. There's resilient children who, in fact, despite exposure to environmental stress, are not necessarily at risk whatsoever. So simply considering environmental risk as a predictor of later mental disorders is not sufficient. So what we're trying to do is to ask the question, can we move under the skin? Can we get a little bit more precise in this? Instead of simply looking at whether children are exposed to environmental stressors, what if we use measures to gauge the biological impact of the stressor on the child, on individual children, and thus identify the kids that are truly being affected by the stressors in their lives? If we could do this, then what we can allow intervention scientists to do is to actually focus their efforts on those children of greatest need. And what we can do is what we do in other areas of clinical medicine is to really be able to identify who is at risk and to bring the intervention to bear to those individuals early in life and prevent the risk for later mental disorder. But to do so, we're going to need far, far better measures of risk in young children.
Well, Michael, it's been a real pleasure talking with you. This is really fascinating research, and thank you so much for your time.
My pleasure, Sean. I hope that was of benefit.
And thank you to our podcast audience for joining us. If you'd like to send us your feedback or suggestions, please send an email to custompodcast, or one word, at aaas.org. For more podcasts in this series, please visit the blog on Learning and Development website by going to bold.expert. And thank you again to Dr. Michael Meaney and to the Jacobs Foundation for making this series possible. I'm Sean Sanders. Thank you for listening.